Hello, my name is Barley Collier. I'm from the University of Oxford and the Plant Sciences department there, and my talk today is there resistance to Hylobius abietis in UK Sitka Spruce? And here you can see me loving my Sitka Spruce. So this talk will be in 3 parts. The first section will be kind of a background, explaining Sitka Spruce, its importance, a little bit about the large pine weevil, which is the common name for Hylobius abietis, and then there'll be a section on an experiment that I recently did and how that experiment was set up, followed by some preliminary results that I have, and then finally we'll finish with what I'm planning on doing next and how the future looks for me. So, Sitka Spruce is the 3rd largest crop by land area in the UK, and economically it produces about £1 billion per year to the UK economy, which is half of all forestry benefits to the UK economy each year. So it's clearly of huge commercial interest. And all Sitka Spruce in the UK initially came from Hylobius off the coast of Canada, because the native range of Sitka Spruce is all along the west coast of Canada, from up in Alaska down through to California. And sites like this picture here are very common in Scotland. Large areas are planted up with Sitka, left to grow for 25 years, and then clear felled to be used for timber or for pulping for paper and various other things. The large pine weevil then is the biggest threat to Sitka Spruce in the UK. Large pine weevil is native to the UK, and the reason that it's so damaging is because the large pine weevil eats the bark of Sitka and a huge range of other trees. But the issue here is that when Sitka saplings are young, they can eat the bark quite quickly, and then they very easily girdle the stems, which can lead to tree death. If the saplings aren't treated every 6 months or so for the first 5 years of their lives, you'll expect around 60% mortality, but up to 100% mortality if they're not treated with those pesticides. Part of the issue here is that the current silvicultural practice around Sitka Spruce, most commonly in the UK, is clear felling, and that causes a population boom for the large pine weevil, because as the site is felled, the stumps release volatiles which attract the large pine weevil to the area. These adult large pine weevils can fly in, they can fly several kilometres. They then land at the site because they've been attracted by the volatiles. They dig their way into the ground, they lay their eggs in the roots of the stumps. These eggs can then take up to 3 years to go through and develop to young adults, which then emerge. And it's often at that stage that the field will be being restocked with now young Sitka. So you've got a huge increase in the large pine weevil population in that specific area because they were attracted during the clear felling, which are now able to eat on any untreated trees. So I'm hoping to see if there's any natural resistance to the large pine weevil in the UK Sitka Spruce population. And my experimental setup was in a greenhouse, and I used young saplings that were 2 to 3 years old, and on each sapling I placed an enclosure. And you can see the enclosure here is attached to the tree with some foam to stop the weevils escaping, and you've got 2 weevils in each cage, 1 male and 1 female. Those weevils are left on for 4 days, and measurements are taken every 24 hours. So the way that I took the measurements for damage is that I'd remove the enclosure with the 2 weevils in it. I'd then wrap the tree in cellophane, as you can see on the left here. I'd draw round in a felt tip the area that had been damaged, the bark that had been removed. I'd then colour that in and take a photograph, and then those images could be processed through Image-J, and that could give you both the total area that had been consumed by the weevils, but also it could tell you the area of each section that they'd eaten, or the average area of the sections that they'd eaten, which can be quite interesting to look at from an insect behavioural point of view, because that will show you how much had been consumed in one sitting, so to speak. So as they sat down and began to eat that time, how much they ate. So I had 13 different families, 20 full sib individuals per family. As I said, I took measurements every 24 hours for 3 days. Well, 3 days subsequently to me starting, which leads to me having 780 photographs to analyse. And so then here are some results. These are very preliminary results. This box plot just shows you with the families, the different families that I was looking at on the x-axis and the area consumed in millimetres squared per gram of weevil on the y-axis. And initially when you look at this, you see there doesn't seem to be much difference between the families. The reasons they're coloured differently is they're from the rooted cuttings that I used were from 2 different nurseries, some from Forestry England and some from Mengmeng. The other thing to note on this graph is you can see that some of the areas appear to be below 0, and that's because the way I'd work out the area consumed on the 2nd and 3rd day was by taking the area from the previous day away. So on the 2nd day, I'd have the area consumed on the 2nd day and the area consumed on the 1st day, and to find out how much had just been consumed over that second 24-hour period, I'd take the 1st days away from it. And so because of some experimental error, some of the areas must have been drawn wrong, because sometimes when you took one away from the other, you ended up with a negative. But you can see that over the 3 days, similar amounts were eaten on each day, area per gram on day 1, day 2 and day 3, looking very similar. You've just got more variation because of those subtractions each day. I've then also done some ANOVAs and some linear mixed models to see if there are any significant differences between families, looking at both the area consumed and the average size of each section consumed. And I've also taken into account various different other variables, including the diameter of the tree, the height of the tree, and various other things that I had measured, the block, which experimenter measured it. But so far, no significant difference has been seen between families. I've also just purchased AS Remel to look at heritability, to see if I can see the amount of area damaged appears to be heritable. But the fact that I've not seen any family effect so far, to me, is not unexpected. As I mentioned earlier, all UK Sitka Spruce is from one area in Canada, Hylobius originally, so I wouldn't necessarily expect there to be a huge amount of variation within the amount of damage here. However, looking forward to what next, I'm hoping to repeat this experiment, but using trees that I've grown from seed from different provenances all across a large section of the native range, so from all of these different red dots shown on the map here. I'm also hoping to repeat the experiment with known resistant and susceptible seed, not resistant and susceptible to the large pine weevil, but resistant and susceptible to the white pine weevil, which is also a bark-eating insect that's very prevalent in Canada, and where they've had huge success with the resistance breeding program to the white pine weevil. And I've got some seed which is known resistant families or known susceptible families that I'm also growing up, which I hope to repeat this experiment with, and also potentially to do a genome-wide association study. So just to finish off, I'd like to thank my lab, John Mackay's lab, and the rest of my group shown here in the pictures, also my co-supervisor Roger Moore, who's at NRS in Scotland, and Forstart, who are my industrial partner.
Hi, I'm Eddie Lauer at the North Carolina State University, and today I'll be talking about broad-spectrum resistance genes in the co-evolved fusiform rust-Pinus taeda pathosystem. My advisor is Dr. Fikret Isik at the Department of Forestry and Environmental Resources. Today's presentation will go in 4 parts. I'll first give you a brief introduction of this pathosystem and some of the genomic mapping work that's been done in the past. Then I'll talk about a QTL discovery experiment. I'll talk about a GWAS validation experiment, and then finally an RNA-seq experiment that was conducted for identifying candidate genes. Fusiform rust disease is caused by a heteroecious rust in the Cronartium genus. This rust can infect multiple species of oak and pine. Pine is the primary host for the rust, and oak is the alternate host. And this pathogen has been co-evolving with southern pine populations for many tens of millions of years, and it causes roughly $100 million worth of damage every year to U.S. pine plantations in the southeast. Previous genomic mapping experiments have indicated that the inheritance of disease resistance seems to follow a gene-for-gene model, or a gene-for-gene interaction, where you have dominant resistance genes and dominant avirulence genes in the pathogen, and when you get specific recognition of a dominant resistance gene to a dominant avirulence gene, the host can mount a disease resistance response and you end up with no disease. So this has been proven by Wilcox and Emerson and a number of other authors working in this pathosystem. And they've always used single strain inocula in these experiments. But I'm a quantitative geneticist and I get to analyze a lot of field data for a living. And the first thing I noticed when analyzing this field data is that parent 10.5, which was used in the original Wilcox experiment, and parents A, B, C, and D, which were used in the Emerson experiments, none of those parents ranked even in the top 30 of our germplasm. So this table here shows a summary of over 600 progeny tests over many decades. And so what I asked was, what is it about parents 4 and 9 that makes them so much more resistant and makes them rank up towards the top? And what is it that gives them such high rank stability across environments? So there seemed to be a knowledge gap to me. So what explains the superior rust resistance of parents 4 and 9 over many environments? Are there single genes that confer general resistance, such as broad spectrum or non-race specific, or just superior marginal effects? So in order to answer this question, we used a QTL discovery experiment using 2 very large crosses of 1,000 individuals each. The first cross used parent 4 as a male, and the second cross used parent 9 as a male. In outcrossers such as pine, we used the 2-way test cross design to analyze QTL data. So in a 2-way test cross, you build separate linkage maps for both parents of a cross, and you use those linkage maps for QTL analysis, genome-wide markers. You can then combine those separate linkage maps into a single consensus map, which was done in this experiment. The artificial inoculation was used to spray basidiospores over seedlings in a controlled inoculation chamber. So on the left, we see the controlled inoculation system at the Resistance Screening Center in Asheville, North Carolina. So we used a bulk inoculum of 100,000 basidiospores per mil. And this inoculum came from 150 distinct rust galls spread across the southeast in these high rust incidence areas. So they were all combined into one single inoculum and sprayed over 3-month-old seedlings. We wanted to use a very high spore density to prevent escapes from the assay. And we wanted to use a very high diversity because we were interested in identifying marginal effects. We're not interested in like specific gene-for-gene interactions, we're interested in marginal effects, so host resistance genes that are effective against a wide diversity of the pathogen. So 7 months after the inoculation, we went back and scored these seedlings for the presence or absence of rust galls. And the picture on the right shows you what a typical rust gall looks like on this would be a 10-month-old seedling. You find these elongated galls with adventitious growth coming off of them. And to the right of that is a non-galled seedling. So this data is recorded as a binary response variable, and it's analyzed with generalized linear mixed models in the QTL framework. So what we found was that on parent 4, there were 2 QTL in linkage group 2 that were separated by about 30 centimorgan, and the resistance alleles at each QTL were linked in repulsion. And we found that these alleles behaved additively with one another, so that individuals that inherited both resistance alleles were completely immune to the disease. On parent 9, there was a single QTL on linkage group 7. So we were able to find that these QTL were all very large effect. The ones on parent 4 each explained more than 20% of the variation. and the QTL in parent 9 explained something like 16% of the variation, so it wasn't quite as effective as either of the QTL in parent 4. This is a picture of the regional linkage map that shows the kind of mapping resolution we have in these populations. We use 1,000 individuals in each population, and we have many, many thousands of markers. So that gives us very, very tight confidence intervals around each QTL. So our confidence interval for each QTL was less than 0.5 centimorgan, which is very high resolution, and it's going to make it easier for us to integress these QTL into germplasm to produce resistant varieties. The second experiment was a genome-wide association analysis to look for these QTL in the field. So this used a clonally propagated population of 2,063 clones from 53 different full subfamilies, and some of those families were related to the QTL discovery populations. This population was grown in 8 environments across the southeast United States, and it was scored at 6 years after establishment for the presence or absence of fusiform rust galls. And then the population was genotyped with the same markers, the P250K array, as the QTL population. We see in the Manhattan plot on the left, sure enough, on linkage groups 2 and 7, the same regions where we discovered the QTL were lighting up in our GWAS. And on panel B, we see a blow up of linkage group 2, where we have 2 distinct regions on linkage group 2 that are suggesting of QTL at these regions. And then on panel C, the QQ plot was suggesting to us that we have very limited LD in the population, so we have also high mapping resolution in the GWAS. The third experiment was an RNA-seq for identifying candidate genes. So this is a bulk segment analysis in which we use bulk samples that were either diseased or non-diseased, and we use needle samples. So if you look at this slide, those 2 circles on the bottom, control, those were random bulks taken before disease challenge, just to establish a baseline of allele frequency or read count ratio for the control sample, just for the population. And then 7 months after the inoculation, we bulked diseased and non-diseased individuals. Each bulk had 50 single needles from diseased individuals or 50 single needles from non-diseased individuals. And we did that for 3 replicates for each disease class from each family. We prepared Illumina libraries from every bulk, and then we prepared PacBio isoseq libraries from the control bulks for the purpose of producing a reference transcriptome. The reference transcriptome, we use PacBio isoseq because it's an assembly-free approach. We did not want to use short read assembly to produce transcripts. We just wanted to get an assembly-free picture of the expressed gene content in each family. The read count ratios were analyzed with a very simple chi-squared analysis in which we had odds ratios. We took the log of those odds ratios and then used Fisher's exact test for testing for associations between the read counts and the disease phenotype. So we found there were, in our most resistant family with parent 4, there were 72,000 high quality SNP. 13 of those, only 13 of those SNP had LOD scores of infinity. So where were those SNP? So there were a total of 46,000 transcripts in the isoseq library for parent 4. Only 231 of those were annotated as NBS LRR, but out of our 13 hits, 2 of those were NBS LRR genes. And those 2 NBS LRR genes clustered closely together in a molecular phylogeny. So on the right is a phylogeny that was produced using the NBARC domain. And so these 2 transcripts had very high homology at the amino acid level, but not quite at the nucleotide level. They're very similar at the amino acid level, so it suggested maybe they have similar function. So putting all this together, we have high-density linkage maps that show that there are genome-wide, there are large effect QTL on parents 4 and 9. We have GWAS that confirms these loci are associated with field-level resistance across 8 wide-ranging environments going from western Alabama all the way up into North Carolina. And we find that these SNP, we find sequence-level associations between candidate genes and the disease phenotype. So this gives us this is a good hypothesis generating exercise, and this might help us with follow-up experiments to validate that these are the genes that are causing the disease resistance in these families. So thanks to the USDA NIFA, Genomic Selection in Forest Trees Beyond Proof of Concept grant, NCSU Cooperative Tree Improvement Program, the USFS Resistance Screening Center at Asheville, North Carolina, and the University of Florida Genomic Laboratory. Thank you.
Hello, my name is Mengmeng Lu. I'm a postdoc at University of Calgary. I work with Dr. Sam Yemen. I'm going to talk about genomic basis of response to Dothistroma needle blight in lodgepole pine. First, I would like to acknowledge my colleagues and funding buddies. A little background about Dothistroma needle blight. This is a pine disease caused by the fungus Dothistroma. The infected trees show red band and needle defoliation, eventually resulting in yield loss and mortality. To study the genomic basis of lodgepole pine response to the fungal pathogen, we first evaluated gene expression in response to the pathogen using RNA-seq. Then we identified candidate response loci using case-control genome-wide association study. Last, we try to validate the candidate loci in the large-scale field using genomic selection models. Our colleagues will finish the last work. In our lab, we designed lodgepole pine SNP array, including disease response and climate response loci. First, evaluating gene expression in response to the pathogen using RNA-seq. This work has been published in the journal Molecular Plant Microbe Interactions. The experimental design was to contrast tolerant versus susceptible genotypes. The trees were inoculated with Dothistroma isolate 1 and isolate 2. These two isolates were collected from different regions in Western Canada. We also included samples inoculated with water as controls. We harvested samples 4 days, 8 weeks, and 12 weeks post-inoculation. We first identified differentially expressed genes, DGs, and genes containing differential exon usage. Most DGs were found in tolerant samples inoculated with Dothistroma isolate 2 4 days post-inoculation. Another peak was found in susceptible samples inoculated with Dothistroma isolate 1 12 weeks post-inoculation. We didn't find many genes containing differential exon usage. What are the functions of these DGs? We run KGG orthology terms enrichment test. This colored bar shows the enriched KGG terms. For example, this lilac bar shows the term cytochrome P450 was highly enriched in DGs. Also, this orange bar shows the plant pathogen interaction terms were highly enriched in DGs. We constructed co-expressed gene modules related to Dothistroma infection phenotypes. We found some gene modules were upregulated, while some gene modules were downregulated compared with controls. For example, these gene module 3 and 4 were downregulated. We checked the genes. We found these gene modules were comprised of photosynthetic genes. This is expected because plant growth tend to be suppressed under pathogen attack. We inferred lodgepole pine Dothistroma interaction pathway, and this pathway showed both PTI and ETI signals can be found. For example, calcium signaling pathway, MAPK signaling pathway, specific disease resistance can be found in this pathway from lodgepole pine. We calculated evolutionary rate of protein coding genes represented by DN to DS ratios using the PAML code ML module. We found in tolerant samples, the evolutionary rate was higher in DG than in non-DGs. We also found the expression levels of DEGs in tolerant samples correlated with evolutionary rate. However, we didn't find this pattern in susceptible samples. We identified candidate R genes with sites under positive selection using the CodeML branch site model. In this table, we showed these candidate R genes, their proportion and the numbers of sites under positive selection. We call these R genes because they contained conserved domains, which are the features for R genes. This is the first part. Please check our paper for details. Second, identifying candidate response loci using case-control GWAS. The plant material was from different regions in Western Canada, including Lodgepole pine, hybrid pine, and the jack pine populations. Trees from each of these 40 sampled locations were planted in controlled environment conditions. Trees in each populations were inoculated with Dothistroma isolate 1 or isolate 2. We selected 5 least infected and 5 most infected trees from each populations for sequence capture and the PUSIC experiment for genotyping. With the genotyped Snips for each population, we run case-control GWAS to detect candidate genes for pathogen response. We first detected genetic structures. This graph shows the populations of Lodgepole pine tended to be clustering together and separated from jack pine or hybrid pine populations. So we run case-control GWAS within Lodgepole pine, hybrid pine, and jack pine populations separately. We only identified significant loci from Lodgepole pine populations. We show these significant SNPs in Lodgepole pine linkage group, which was detected by Tegan MacDonald in our lab. This Manhattan plot shows the candidate loci and the candidate regions identified from different Lodgepole pine populations. These candidate genes include R genes, transcription factor genes, and the translation initiation genes. We also found in candidate genes, the linkage disequilibrium was higher than that in non-candidate genes. This suggests these significant loci may be driven by selection. This is the second part. Last, I would like to introduce Lodgepole pine SNPs array version 2. We call this version 2 because in this SNP array, we included new probes based on fungal pathogen response SNPs identified from this project. We also included old probes from our previous project, which have been verified. Most probes can be anchored on current linkage groups, as shown in red bars. We hope this Lodgepole pine SNP array version 2 will be a useful tool for genomic selection. Thank you for listening. Any questions?
Hi everyone, my name is Samson Osadolor, and I'm a PhD student from the University of Alberta. Today I'll be talking about my project, which is on Lodgepole and jack pine molecular responses to Western Gall rust. Western Gall rust is a disease caused by the rust fungus Cronartium Harknessii. It's a disease that leads to the production of galls on stems and branches of plants. This disease affects trees of all ages, but young trees tend to suffer more through severe deformation and sometimes mortality. In Canada, the main host of the fungus are lodgepole and jack pine, and studies have shown that jack pine are relatively more resistant than Lodgepole pine in response to the fungus. Currently, efforts have been made to breed for resistance against Cronartium harknessii, and part of this effort involves trying to understand how defense responses to Cronartium harknessii differs in more resistant and more susceptible pines. And so to understand how responses to Cronartium harknessii differs, first we need to understand the general plant defense mechanism against pathogens. Pathogens are divided into biotrophs and necrotrophs based on their lifestyle. And so biotrophs, they feed and reproduce on living host tissues, while necrotrophs, they kill host tissues for nutrition and reproduction. The detection of these pathogens by the plants induces signaling pathways that are regulated by defense hormones, which in turn activate defense responses. And so the general model is that attack by biotrophs induces a defense response that is mainly regulated by salicylic acid, while attack by necrotrophs induces a defense response that is mainly regulated by jasmonic acid and ethylene. And between pathways regulated by these hormones are crosstalks. So these are actually interactions that can be antagonistic or synergistic. And some pathogens try to use this crosstalk to their own favor to suppress the defense of the host plant. Rust fungi generally are considered obligate biotrophs, and so we expect a biotrophic interaction between Cronartium harknessii and Lodgepole or jack pine. Since salicylic acid is the main player in this interaction, we expect higher levels of salicylic acid in pines that are more resistant to Western gall rust. As I mentioned earlier, jack pine are relatively more resistant than Lodgepole pine in response to Western gall rust, and so we expect greater induction or earlier expression of genes associated with Western gall rust resistance in more resistant jack pine than in more susceptible Lodgepole pine. Similarly, we expect greater induction or earlier expression of genes associated with Western gall rust resistance in more resistant Lodgepole than in more susceptible Lodgepole pine. So our objective is to determine how defense gene responses and also defense hormone accumulation, how it differs between more susceptible Lodgepole pine and more resistant Lodgepole pine. Also how it differs between more susceptible jack pine and more resistant jack pine. And also how it differs between more susceptible Lodgepole pine and more resistant jack pine. We're using three approaches to achieve these objectives. First is resistance screening to verify the phenotypic responses of the pine family used in this experiment. Second approach is gene expression analysis, and we're using both RNA-seq and QRT-PCR. Our last approach is hormone quantification, and here we use liquid tomographic mass spectrometry. To screen for resistance, we carried out a growth chamber experiment that includes 6 pine families, 2 jack pine families, and 4 Lodgepole pine families. Resistance of these pine families have previously been characterized from field assessment and also growth chamber studies. So we wanted to verify their phenotypic responses by comparing percentage of seedlings that produce galls following inoculation by the fungus. Here we can see that the more susceptible Lodgepole pine family produce more galls than the more resistant Lodgepole pine family. Similarly, when we compare the jack pine families, we see that the more susceptible jack pine family, that is JP2, produce more galls than the more resistant jack pine family. And overall, we can see that jack pine families produce less galls than Lodgepole pine families, which also confirms that jack pine are quantitatively more resistant than Lodgepole pine in response to Western gall rust. To compare defense gene responses, we are making use of RNA-seq data, which was generated from previous Western gall rust experiments carried out by Chandra, a former postdoc from our lab. This data was analyzed by Rhiannon and Juan, who are currently postdocs in our lab. And so RNA-seq helps us to examine the transcriptome. So here is the expression of all genes that are induced in response to the pathogen. Also, it helps us to examine the differential expression of genes by comparing more can-inoculated samples. QRT-PCR does the same thing, but with QRT-PCR, we can only analyze the expression of fewer genes that we already know. And we are using QRT-PCR for validation of key genes identified from the RNA-seq dataset that are known to be involved in the defense response. Here I'm showing you genes that are putatively involved in the biotrophic response based on what we know from model plant systems like Arabidopsis. So genes involved in salicylic acid biosynthesis as well as its signaling. The heat map here indicates the differential expression of genes based on their log4 change. So we can see that genes involved in salicylic acid biosynthesis and signaling are not differentially expressed, although the defense response is clearly activated, as we can see from upregulation of pathogenesis-related genes. Now, what's also interesting is that when we compare Lodgepole and jack pine, we can see a more intense response in jack pine than in Lodgepole pine for both time points. At the moment, we don't know the pathway that is activating the defense response. Clearly, it's not salicylic acid. I've also looked at the pathway regulated by jasmonic acid and ethylene. So here I look basically at the genes involved in jasmonic acid biosynthesis, and here we can see that they are more differentially expressed in jack pine than in Lodgepole pine. I've also examined the hormones to observe their contribution to defense responses. So here we compared half-seed families of jack pine and Lodgepole pine that are known to be more resistant or susceptible to Western gall rust. So following inoculation, we examined levels of salicylic acid as well as conjugated salicylic acid, which is an inactive form of salicylic acid. We also examined jasmonic acid, which, unlike salicylic acid, is inactive in its feed form, but active only when it's conjugated with isoleucine. And so we predict higher levels of salicylic acid following attack by the fungus. As we can see, there is really no clear trend in all the time points that suggests that salicylic acid is being induced in the defense response. So when we compare all the pine families and also all the time point that is 3 days post-inoculation, 7 days or 14 days post-inoculation, there's really no clear trend that indicates that salicylic acid is involved in the defense response against Cronartium harknessii. Similarly, when we compare conjugated salicylic acid, although I'm not showing the graph here, we didn't really see any trends or patterns that indicate their contribution to the defense response to Western gall rust. Now, jasmonic acid is usually not involved in the defense response against biotrophs, but we found that jasmonic acid is induced by Cronartium harknessii, particularly when we compare the most susceptible Lodgepole and the most resistant jack pine family at 7 days post-inoculation. We see that the most susceptible family in Lodgepole has more jasmonic acid than the most resistant jack pine family. When we compare the bioactive form of jasmonic acid, that is jasmonic acid isoleucine, we didn't see any change that indicates their involvement in the defense response. So far, our results from resistance screening have shown that jack pine are more resistant than Lodgepole pine to Western gall rust. This more resistance in jack pine correlates with more intense response, as we see from the expression profile of the defense genes. Lastly, from our hormone analysis, we believe that the induction of jasmonic acid may be lowering the amount of salicylic acid through antagonistic crosstalk. So in conclusion, the different patterns in the expression of defense genes in response to Western gall rust when we compare Lodgepole and jack pine indicates more resistance in jack pine than in Lodgepole pine. Our results also show that salicylic acid signaling is really not a key part in the defense response to Cronartium harknessii. And also, analysis of jasmonic acid and jasmonic acid isoleucine levels suggests that the pathogen might be inducing the synthesis of jasmonic acid to suppress the salicylic acid signaling pathway. Hopefully in the future, we can dissect the molecular mechanism underlying the antagonistic crosstalk, as well as other pathways that might be involved in the defense response. To wrap things up, I would like to thank my supervisor, as well as other members of the Cook Lab, both past and current members, who have been very helpful every step of the way in the progress I've made. And also, I would like to thank the funding agencies for their financial support to the project. Thank you all for listening as well.
Hello, My name is Heather Dunn and I'm a PhD student at the University of Oxford in collaboration with Forest Research. My work looks at sudden larch death and the variation in host response to infection. Sudden larch death is caused by Phytophthora ramorum, which is better known in America as the cause of sudden oak death. In both cases, ramorum causes bleeding lesions on the trunk that can girdle the stem and lead to death. However, in larch, there is also high levels of sporulation in the canopy, leading to rapid infection spread and whole stand mortality. As you can see here, in 2012, all of these bright green trees are healthy larch, and then 6 months later, in 2013, there was a high level of mortality throughout the entire hillside. Larches are the third most important conifer crop in the UK, and we have 2 species in commercial production, European larch and Japanese larch. European larch was suggested early on in ramorum infection to be less badly affected than Japanese larch. However, this might have just been due to the small area of European larch planted, as it only accounts for 15% of the total larch forest. The objectives of this work were to investigate the differences between European and Japanese larch. We decided to do RNA transcript analysis to look at the accumulation profiles in response to infection, identify the molecular interactions between the host and pathogen in order to identify differences in response between European and Japanese larch. This could then help answer the question of possible greater resistance in European larch to ramorum infection. We carried out inoculation trials at 4 time points with 8 trees per treatment combination in a randomized complete block design. We inoculated into wounds in the bark of the main stem and then bagged the trees after inoculation to increase humidity and the successor infections. A subset of trees were destructively sampled at each time point at 1, 3, 10 and 25 days after inoculation. And you can see here a small lesion that has developed at 3 days post-inoculation. Samples were then taken from the margin of the lesion, just here, for RNA-seq, and a subset of 3 trees per treatment combination was sent for poly-A enrichment sequencing at the Oxford Genomic Centre. We then carried out differential expressed gene analysis in DSeq2 to create respective gene lists that were differentially expressed by time, treatment, and the interaction between them. We also carried out hierarchical clustering on the differentially expressed genes and the time by treatment interaction to show expression profiles over time. And you can see here the difference between various genes in the control and inoculation treatments. When we looked at lesion development, you can see that there is no lesion development on control trees, and there was no lesions observed at 1 day post-inoculation. Lesions then increased in length over time, but there was no significant difference between the 2 species at any time point. If we look at the gene expression data, we can see the clustering of different samples. On the left here, we have principal component analysis of the overall set of gene expression data, which indicates a clear separation between the control and inoculated treatments. However, we can see that some of the inoculated treatments cluster with the control trees, but these are from the first time point when there is no lesion development, suggesting that the response to infection had not been activated at this time point. If we look at the right-hand side, we have the same analysis, but the shapes refer to the 2 different species, and you can see there's European and Japanese larch and no clear separation between the 2. If we look at the lists of differentially expressed genes for each species and compare them to the annotated reference transcriptome of Larix laricina in order to identify corresponding tear annotations. These tear identifiers were then used to look for the enrichment of gene ontology terms. We can see that there is an overrepresentation of genes related to jasmonic acid biosynthesis in both European and Japanese larch, with the same 15 genes being significantly differentially expressed in both species. However, there are also more functional groups relating to jasmonic acid in the European larch, with regulation and response genes also being overrepresented. Jasmonic acid is a signaling molecule which activates and regulates other defense pathways, so an enrichment of the jasmonic acid-related pathways suggests a strong defense response. If we look a little bit closer, we can look at LOX genes, which are well characterized for their role in rapid jasmonic acid synthesis after wounding and in response to infection. The expression profiles of the LOX1 homologs show differences in gene expression between European and Japanese larch. In European larch, we have rapid induction between 1 and 3 days, followed by sustained or increasing expression towards the end of the experiment. In comparison, Japanese larch has larger increases in some homologs, but this is more transient, and it starts to decrease towards the end of the experiment. We can also look at the KEGG pathways. There are several enriched pathways that are likely to be involved in defense response, including the biosynthesis of antibiotics in both species, which includes the biosynthesis of tropane, piperidine and pyrimidine alkaloid, terpenine backbone, and isoquinoline alkaloids. European larch also has enrichment of the biosynthesis of secondary metabolites, which includes the phenylpropanoids and the flavonoids, the terpenoids, and the alkaloids. Both species had enrichment of the flavonoid biosynthesis processes. Flavonoids are important secondary metabolites in defense response. We saw similar differences in expression profile between the two species for the flavonoid biosynthesis genes, with European larch having expression profiles that showed rapid increase in expression, followed by sustained or increasing levels of expression to the end of the experiment, whilst Japanese larch had the rapid but transient expression decreasing towards the end of the experiment. If we look at genes involved in the flavonoid biosynthesis pathway, such as chalcone synthase homologs, we can further investigate this. Chalcone synthase catalyzes the first step of the flavonoid biosynthesis process by directing carbon flux from the general phenylpropanoid metabolism into the flavonoid pathways, and alterations to the chalcone synthase expression are thought to affect the accumulation of flavonoids, with increased levels of chalcone synthase shown in other cases to increase the activation of flavonoid biosynthesis and improve resistance of conifers to fungal pathogens. We can see here that there are remarkably similar upregulation profiles across all homologs, which suggests that the accumulation of extra chalcone synthase genes is under positive selection. If we look at another family of genes, in this case the nucleotide binding leucine-rich repeat genes, which form the largest resistance gene family in plants and are the main group of cytoplasmic receptors involved in the recognition of specific pathogens as part of effector-triggered immunity, we can see that there is a large number of genes in both European and Japanese larch. Almost all of these are downregulated, which is what we would expect because these genes tend to be specific to specific pathogens. In this case, we have two genes of interest, these two here in European larch, which are increased in expression in response to pyrogen infection. This could be of use in further work, as it suggests that there are some of these genes, particularly in the TIR subclass, which could be specific to pyrogen infection. In conclusion, although there are no differences in lesion development, there were differences in gene expression. European larch has rapid induction of defense genes, followed by sustained or increasing expression, whilst in Japanese larch, the observed gene expression increases are larger but transient, suggesting a strong but short-lived response to infection. This variation in response to infection could help explain the differences seen in the field conditions and should be considered in the selection of trees for planting and future breeding. I would like to thank my lab mates at Forest Research, Sarah, Carolyn and Debbie, and at the University of Oxford with John and Henry, and particularly Henry for his help with the DE-seq analysis. This work was carried out with funding from the Scottish Forestry Trust and Forest Research. Thank you for listening. Are there any questions?